So good morning, all of you. So today I'm going to do the part of the biology, which is most probably interconnected for your um, O-level syllabus. So before going to start, because I want to thoroughly know about, please don't unmute yourself because you have to keep your microphones always muted. So there is any interruptions because we will upload these recordings in the YouTube. So that's why I wanted to do the before beginning my lectures. You have I will couple of information. So anyway, so because just when you come out the biology part, because you have to understand the basic fundamental about the mainly cell structure and functions. So basically, you have to thoroughly understand the structure and function of the plant and the animal. So when you come out the biological surrounding worlds, because you have to native for the biological plants as long as microorganisms and also animals, those are main composition ingredient. I will be cover later on what is mitosis, and the nitrogen circle and the structure of the plant, the involvement and vegetative propagation and the self-artificial pollination and the self-pollination, cross-pollination and the physical effect. These are the every interconnected your structure paper. So more or less similar. I will be assured on this before clarifying for all, right? So when you promote the cell structure and function, it is mostly like defining the paper in the structure paper, right? So most probably will be appeared on the structure paper. When you go through about your past papers, just you can imagine that if you can determine the some kind of the terms and the norms in the past paper, last time they didn't might ask about the cell structure and function part. So you have to thoroughly understand this time, sometime the figure will be given in the paper. So, and you have to thoroughly understand what are the specific terms and what are the specific coordinate parts which is connected in the plant tissue and the animal tissues and the microbial cell. Then you have to thoroughly understand each and every point, and you have to determine the each function very precisely, because when you do the curriculum for the A-level biology, biotechnology, because you have a fundamental knowledge. That's why I included. So before moving the lessons, because just you can see the introduction part, it is mainly overview about the introduction. So in the back into 1665, the Robert Hooke observed a section of a croc using a microscope prepared by him. So continue of the cell theory. This is a very important, dear students. When you came out for the cell structure, you have to came out this part. It is very important and interconnected. You have to determine the thorough idea. So first content of the theory, the cell theory, what is this? Because this is very important. You have to understand sometimes the MCQ they ask in A, B, C, then you have to select the proper phrase and the proper statement, then it is interconnected with the cell theory. The first one is you can understand the structure and the function is of the cell. So basically, I will give the more or less similar, and I will give the thorough information. So I really recommend all of you to take out your note, and while I'm lectures, you have to, while I'm lecturing, you have to take out your own note and you have to prepare your own self, right? So the first one is the cell theory. What is explained that the structure and function unit of a life is a cell. So when you consider about the animal, it can be either plant, it can be a microorganisms. You have to understand basically atom. It is a molecule structure of the atom. Then that atom will be grown up as a particles. That is mainly from the our body, right? So first one we have to use the atom. That atoms make some kind of the molecules. That molecule interconnected with the interconnected with the some kind of the large molecule, then they are make the cell. That cell make the tissue makes the organ make the animal. Likewise, you have study basic things, right? So before move on these, you have to thoroughly understand the structure and the function of it. So the first thing theory saying the structure and function of a unit of a life is a cell. Yes, we all know we all are made up of single cell or a multicellular. Because we are an animal, no, you can sometimes see you can yeast. You can see it sometimes yeast. Yeast is a mostly Saccharomyces cerevisiae. This yeast Saccharomyces cerevisiae is mainly denoted with a single cell, right? So you have to mark this is a single cell. This is a fungi. Can be appear in your MCQ papers, right? So you have to take out your own note, and I'm give the gentle reminder once again. So dear students, first one, you can see the structure and function unit of a life is the cell. That is we already know. Second one is, you can see the second one is mainly it is the main part. All the organisms are made up of one or more cells. I already mentioned about that. So because when consider about the cells, that cells are made of one or more cells. It can be either diploid, it can be either haploid gametes. Sometimes maybe you have to thoroughly figure out that, because sometimes some organisms are one cell, some organisms are two cells. Thus, I already mentioned that yeast, it is a scientific name Saccharomyces cerevisiae. You know already that you can learn about these things during the fermentation of the bread and some kind of the beer and all the alcohol. Mostly this is Saccharomyces cerevisiae, very important. So it is made from the single cell, and also you can see the mainly organisms have one or more cells. This is a second phenomenon you have to take a remember, right? Most probably students are not going to look these things, but it can be asked various struggling the questions during the, they can be changed the pattern. Likewise, this is a format, A, then this is a B, then C. They will change some kind of the things, but you can determine, okay, this is correct, then you can put that answer. Then I will for the first one, the structure and function unit. Life is a cell. The cell is a basic fundamental for the all the human beings, all the animals, all the plants, and also all the microorganisms. The second one is all the organisms are made up of one or more cells. The third one is new cells are formed from the pre-existing cells. That is very important, because we are going to learn about the mitosis and meiosis. Just it is interconnected with the pre-existing cells, because just determine in the father and mother, because the father is a can be make of the sperm cell, this is a mother can make for the egg cells. Both are in the fertilization step, right? That fertilization embryo came to the some kind of the making the cell wall and the ovary part, that is also pre-existing of the cells. How can baby make? So how can the humans are arise? How can be animals can be determined for the world? You can just consider about the three term. New cells are formed as the pre-existing cells. And these are very important key point. Just you can write it down, right? I can give you just 1 or 2 minutes. Then you have to keep it now, because we have a long part in the discussion about the every each and part of the animal cells and tissue culture component and the self pollination and the artificial pollination. If you're unmuted, please turn off your mute, right? Okay, next one, I'm going to make you concept of the cell. Concept of the cell. What is the main concept of the cells? That is very important to understand while you are slotted out for the plant cells and also animal cells. So just you can understand what is the concept of the cells. The cells are the smallest structure unit of an organization of a living body. That is very important. It is a very small structure. How you can see the cells? That is one of my questions. When you do the some kind of the experiment, it can be some kind of adipose cells, subcutaneous cells. It can be a sometimes red blood cells and the white blood cells and the plates. You can determine those structure from the microscope. It can be a light microscope or else it can be a compound microscope. You can view those structures. The part of that, so you can see here, the cell is a smaller structure unit of an organization of a living body. That is very, very important point to you have to keep in remember. The cell is a smaller structure because sometimes you may think, oh my God, I know the cell. Why sir is teaching? Because most of the paper evaluation says lot of students know the not much about the basic fundamental in the cells, because animal cells and the plant cell, those are very moment basic structure in your paper, and the life also as well. The second one, you can see the organisms that compose single cells are called unicellular. What is unicellular organism? Give me some example. Unicellular. You can use the chat box if you prefer. What is unicellular organisms? I already mentioned that. The yeast, Saccharomyces cerevisiae is the unicellular organisms, and some multicellular organism, likewise the animal, plant, some microorganisms. Those are mainly multicellular organisms. So instead of we are moved to that, so you have to thoroughly understand a small specification about the bacteria cell. So this is, I'm going to write here the small structure of the bacteria cell. So this is a structure of the bacteria. You can see some kind of the outer layer here. So mostly it have a cell wall. It is made from the peptidoglycan. You can take it the note. This is a bacteria. This is a bacteria. Bacteria cell wall made from the peptidoglycan. Bacteria cell wall is made from the peptidoglycan. So you can see some kind of the cytoplasm. You can see cytoplasm, and also you can see the round plasmid or their DNA, and you can see the Golgi apparatus, or else you can see some kind of the intracellular matrix, right? So then you can see the capsiums, you can see some kind of the capsiums, and you can see some kind of the fragment. It is held for their reproduction structure. This is mostly held for their reproduction system. So it is can be interconnected with the another bacteria organism, and they are doing their reproduction, and they are doing some kind of the diploid gametes for their surrounding and their future movement. This is a basically structure of the bacteria. You have to know what is the cell wall. Then you can write a note. Cell wall is made from the peptidoglycan. Peptidoglycan. That is very important. Third one, you can see ribosomes. They are protein synthesis. Ribosomes, they are protein synthesis. Then you can determine the DNA or a plasmid. It consists their genetic information. You can say they are consist with the genetic information. And also when you came out the fungi, because I here mentioned two main typically of the cells, that is plant cell and the animal cells. Just you can see, I'm going to use my whiteboard. So you can see first one is mainly the fungi or a fungi or fungi. So that fungi is mainly basically consists with the humans as the animal plant cells. It is the eukaryotic, not the prokaryotics, right? So when you can determine as there is a cell type, there is a cell type is, because I'm not going to put in all the details in the slide. While I'm lectures, I'm do my extra on words and I'm doing the extra lectures. Then you can write it down, right? You have to take out your note. So the cell, this is the mainly interconnected with prokaryotic cells and the eukaryotic cells, prokaryotes and the eukaryotes. This is very important to know these terms, right? So if you prefer to A-level doing the biotechnology stream, you can anymore contact me. Otherwise, you have to do the biology stream, you have to contact me as well. Then you can definitely understand these basic fundamentals are very important. You have to understand the basic things of the biological concept. So this is the prokaryotes, this is the eukaryotes, and the prokaryotes, it is a, don't have proper structure, right? It don't have a proper structure, but that is not consist with the, you can cite that is no proper structure, no proper structure, and also it is don't have the cell, it don't have the organelles. You have to write, it don't have organelles that are composed with the, they are composed with the, they are composed with the cellular matrix. Because when you see about the some kind of the mitochondria and vacuoles and the Golgi, sometimes nucleus, nucleolus, Golgi apparatus, those are mainly not composed with the layers. Just you can see the prokaryotes, they are just this a structure. But when you come to the eukaryotes, when you come to the eukaryotes, it have a quite nice shape around the outer layer matrix. It is composed their cells and the structures. So just you can hear example prokaryote bacteria, cyanobacteria, cyanobacteria. This cyanobacteria is photosynthesis. This can be photosynthesis. So it is heterotrophic or autotroph. It is an autotroph, right? It is an autotrophic because it can be produced their own by the photosynthesis. So this cyanobacteria is mostly help in the nitrogen fixation. Nitrogen fixation. So it can be a free living, it can be a free living nitrogen fixation. So then I think you are clear about the what is prokaryote. Prokaryote have a don't have a proper structure, right? So when you consider about the bacteria, some kind bacteria is consist bacillus, pseudomonas, coliforms. Those are different structures. We cannot predict this is the same structure like animal structure, plant structure. Then it is not composed with the layers, that is not consist with the outer layers, that is embarked with the cellular components. Then the second one is the eukaryotes. It is the mainly contact with the organelles, that is bounded with the cellular matrix. So eukaryotes, you can see animal cells, plant cells, those are mainly, and the eukaryote and the prokaryotes and the protozoa, protozoa. So those are mainly come for the algae. Algae also similar structure, right? So that is composed with the mainly eukaryotes. Then I think you are clear about what is the mainly basic fundamental about what is eukaryote and the prokaryote. So I will move on to the typical cells of a two cells you can determine, that is the plant cell, animal cell. Just you can understand what is the animal cell and what is the plant cell. This is very important. So why I wanted a question from you, why plants look like as a green? Just plants, it is a green color. If it is a sky, it is a blue color. If it is a soil, it is a brown color. That is your theory, you learn. I'm asking that what is the scientific reason behind it? Just you can see the plants. When I see the plants, the surrounding environment, that plants are mainly green color. Why is that? Yes, very good. Exactly answer I received from the Haya. Thank you very much. Due to presence of the pigments called chlorophyll. Yes, excellent, Fatima. So I received both answers. Thank you very much. Excellent answers. Because when you consider about the plants, that plants are of green color because of the pigments called chlorophyll. That pigment, plants have a pigment, that is the chlorophyll, chlorophyll A, chlorophyll B. We can divide up chlorophyll A, chlorophyll B, anthocyanins, right? Anthocyanins. Because seminar is giving the more of a knowledge and the expertise that these field. So I'm the lecturer at a Mercy Education campus. So we are giving the much more thorough idea and the deep consecutive for your enhancement. So you cannot expect this is a school and that this is not a tuition, right? So you can see the typical cells around the plant cell and the animal cells. So then I will move on to the next plant. What is a plant cell? This is a very, very important to learn about. Definitely you have to go through these, and I know how the papers going to be exposed, and I know how to papers going to be prepared. Your papers already been prepared by the professors and the university lecturers, so you can understand science is very easy for subject, but students are looking as a very hard subject. So when go to the schools, lot of schools are not obtain a resource. Very least amount of the students are got about the science A. When I meet my students, they are say sometimes they might be saying very, very hard science paper, but it is not a very hard. It is a very easy subject, so you can properly study. I'm going to move for the plant cell. When consider about the plant cell, it is a very important to understand the specific terms. First, it is a very important about the cell wall. You can see here, I have marked here cell wall. You can see this is a cell wall, right? This is a cell wall. What is the main component made about the plant cell wall? You can write cellulose, 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 lignin, lignin, and the pectin. You can see the both main structures of a unit plant cell is very important. So dear students, the when consider about the cell wall of a plant have a cell wall, it is a give the protection layer. So what is our protection layer? What is the cell wall for us? Like a skin, right? Not a skin. I'm just making it as an example that skin can provide unusual substances, microorganisms to insert about the inside of a skin part. It can be a dermis, epidermis, right? So then cell wall is made from the mainly cellulose. The second one is lignin. Third one is pectin. These are very important, right? So why we can't digest the cellulose in our body? Why we can't? Can we eat some kind of the leaves? Can we eat some kind of the leaves? If can, but it cannot be digested because some kind of the cellulose material, when in go through the inside of a body, there is no any enzyme to digest the cellulose. You know, the cellulose can be emitted, it can be break down the simple molecule as a glucose, right? Glucose, the involved the enzyme is cellulase enzyme. What is the enzyme? Cellulase enzyme. That cellulase enzyme, they cannot disrupt, they cannot break down the particles which is interconnected for the cellulose to small particles. It is can be either glucose molecules, most probably. It is the enzyme is interconnected cellulase enzyme. Then this is the structure, schematic diagram of the structure. You can see this is a cytoplasm. Then I say that first one is cell wall, cell wall. The second one is cytoplasm. Still likewise the core gel structure. Third one is cell membrane. You can see third one is cell membrane. You can see here this is a cell membrane. And you can some kind of the ribosome. This is the ribosome. The ribosome. And Golgi apparatus. It is very important. This is a Golgi apparatus. Then you can do the chloroplast. This is a chloroplast structure and the chloroplast. And this is a nucleus. You can see this is a nucleus and nucleolus. It consists with the most fundamental of the genetic materials. And also endoplasmatic reticulum. This is endoplasmatic reticulum. It is a smooth and the rough endoplasmic reticulum. This is a mitochondria. This mitochondria is always, we are say, is the power of a cellular unit, right? It is always can be produce some kind of the energy. Third one is ribosomes. Fourth one is vacuole. Fourth one is vacuole, right? The mainly fourth one is vacuole. You can see it is a very important to understand the structure of the vacuole. So this is a vacuole part, right? So vacuole have some kind of the unsaturated liquid, suspended, and the solids particles. Then we are moved for the animal cells because this is very important, dear students, because if you are going to thorough this biology part, then you cannot need to go the beyond for these lessons once again. You can directly go for the exams and you can write. Here, the second one is mainly you can see some kind of the here cell plasma membrane or a cell membrane. Then this one is a mitochondria. You can see this is a mitochondria, then peroxisome and rough endoplasmic reticulum and the ribosomes. You can see ribosomes have a protein synthesis. Give me a 1 minute. Okay, right. All right. So next one is you can see the vacuoles and the mitochondria and lysosomes, peroxisomes, cytoplasm, and Golgi apparatus, smooth endoplasmic reticulum, and the rough endoplasmic reticulum, nucleolus, chromatins. These are very important when you do the molecular biology, molecular genetics, immunology, and also mainly in your terms in the cell structure and function, it is a level second unit, right? Biology, right? So now let's move to what is the different core differences between animal cell and the plant cell. This is a animal cell, this is a plant cell. You need to must know about the basic fundamental differences. So the first one is you can see the cell wall present. Animal, it is not cell wall is not present. So what is the cell wall made from the plant cell? I already mentioned cellulose, not a cellulase. Cellulose. Some students are writing the exam paper cellulase. It is wrong. So plant cell, they have a cell wall present. Animal cell, they have a cell, they don't have a cell wall. The second one is cytoplasm is a push towards the one side. The cytoplasm is pushed toward the some kind of the corner side. Large content of the cytoplasm content when consider about the this is a structure of the animal cell, this is a overall on the cytoplasm. But when consider about the cytoplasm is a toward of a push. It is a not here vacuole, you can see some kind of here, this is a push from this part. That is they are explaining. The third one is a large control vacuole is present. You can see I already mentioned this large amount of the vacuole. That vacuole is the imbalance of a nutrition cycle. You have to write. It is a very important for the balance of a plant nutrition cycle, metabolic activities. Right. The next one is large content of the cytoplasm that is present. Third one is mainly large amount of vacuole is absent. Can you see the vacuole in the animal plant? Can you see the vacuole in the animal plant? Not exactly, you cannot see, right? Then chloroplast, it is the mainly consists with the chlorophyll substance. It is mainly consists with the chlorophyll substance, but it is not present in the animal cell. It is absent, but here chloroplast is present. Okay, then I will quickly go to the one by one. Just you can see cell organelles in the structures present in the cell. That is very important to understand. The first one is cell wall. You can see this is a cell wall, and it is the outermost covering of a part of a cell wall. It is a mostly outer layer part. It is a mostly, you can see, it is a basic outer layer part. Is a basic outer layer part. It is a dead structure. It is always dead structure. Some kind MCQ they may be asked. The main constituent of a is cellulose, right? So that is very important. And function, mainly function that maintain the shape of a cell and support and the production of the cell. Because I will be discuss about the nitrogen circle later on, and I will go some biosphere. So you can stay from here. So next one is plasma membrane. What is the plasma membrane? The plasma membrane is present interior to the cell wall, and it is mainly present in the interior to the cell wall, right? These are very important to key terms. When you come on the plasma membrane, mostly it can be found in the animal cells, and as long as it is present in the plant cell as well. So plasma membrane is present interior to the cell wall of a plant. When consider about the plant structure, this is a plant cell wall, then you can determine in their outer part layer, from inside layer, it is a cellular membrane. Cellular membrane or a plasma membrane, both are same terms. The boundary of animals is a plasma membrane. It is made up of phospholipid and the proteins. Very important. Sometime in the MCQ ask how, what is the main molecule that is made from the plasma membrane? What is the main molecule? Phospholipid. If just assume that if there is a component or a food that is consists with some kind of the lipid, how you can determine the concentration, how you can determine the amount of the lipid, what is the test you can use, what is the test you can use for the lipid analysis? If there is a present of the lipid in the cell, Sudan 3, right? You can use as a Sudan 3. So that is a very important. And just we can quickly go through the function. Yes, very good, Haya. So she is the only one students giving the answers. Other students also have a try, right? Function, it is the enclosures of a cell allowed to entry of a water, ions, and some molecules, and thereby control the entry of exist of the materials into the outer of the cell. That is very important to function. It is the enclose of a cell. I already mentioned about that. So it is enclosures of a cell. It is enclosed. It is enclosed. Just you can see this kind, sometimes this may be, this is the enclose of a cell and allowed to entry of a water, ion, and some molecules thereby and control the entry. It is allow for the entry and existing. Likewise, our home door. So next one, cytoplasm. Next one, you can see what is the importance of a cytoplasm. So cytoplasm is one of the structure. Cytoplasm, it is mainly gelatinous liquid part of a cell. It is likewise a jelly-like a substance. I already mentioned that. And including organelles as known as a cytoplasm. That is a jelly-like a matrix. Like you can write a small term here, the cytoplasm. When you keep in remember any terms and the specification, the cytoplasm is a mainly jelly-like a structure. It is a jelly-like a structure. So function, it is a mainly function about maintain the shape of a cell. It is a very important for the cell structure. This is like an the structure. If there is a cytoplasm, there is, if just assume that there is no any cytoplasm, how about the cell structure? It is a likewise a bread or something else, right? It is a likewise a bun, but you can see the maintain the shape and where the cell organelles and carry out different metabolic pathways, because just you can see when our body, it is so many of the metabolic activities can be present, and the some kind of the extraction can be takes place in the body. So next one, some organelles are surrounded by the cell membrane. Some organelles surrounded by the cell membrane. The first one, cell membrane, is a mitochondrion. The second one is nucleus. Third one is endoplasmic reticulum. Fourth one is Golgi complex or as Golgi apparatus. Just you can imagine that here mitochondria, it is mainly important for the cellular respiration. Nucleus, it consists with the genetic information. The third one is endoplasmic reticulum. The fourth one is Golgi complex or as Golgi apparatus. So that is the organelles. In the quite MCQ, they may be asked, what is the organelles? Can you specification cell membrane? These are I already made cell membrane eukaryotes type. Eukaryotes, not a prokaryotes type, because they compose with the cellular membrane or a intracellular matrix. The fourth one is nucleus. Nucleus is the main organelle in the cell. That is very important. How we are born for the world. It is also one of the activity of the nucleus, right? So because in there is a male female part that can be produced in the reproductive systems and the reproductive parts, then we can be born. But we have also nucleus in the cells. Nucleus is the main organelle in the cell. It is surrounded by the nuclear envelope. It is it is covered by the nuclear envelope, and also one or two molecules and a chromatin body present inside of a nucleus. Chromatin fibers, that is very important. They are made the chromatic body convert into the chromosomes. Nucleus, when considered about the nucleus, it is considered nucleolus, that is have some kind of the chromatin fibers that can be made in a chromosome. How many chromosomes present in our body? There is a 46 chromosome in the 23 pair. How much of the chromosome present in the rice? How much of the chromosome present in the rice plant? There is a 11. In the fruit fly, there is a 17 chromosomes. You have to mark this is a rice plant. This is a fruit fly. So what is the function of the nucleus? The storage of genetic materials and the transfer the inherited characters from the generation to generation. It is a very important to storage the genetic materials and the transfer the inherited characters from a generation to generation. Mitochondria. Mitochondria is a cellular respiration. It is the take place of the cellular respiration. You have to write the mitochondria. It is a cellular respiration. That is very important. It is the term denoted in the photosynthesis. You know photosynthesis. Where the photosynthesis are take place? Where the photosynthesis are take place in the yes, exactly. Because when you consider about the chlorophyll, right? So chlorophyll pigments have, yes, in the plant, Shafia saying in the plant, yes, we know. In the plant, which structure photosynthesis take place? You know, it is a very basic. Yes, chloroplast, right? In the chloroplast, that is photosynthesis can be make place. That photosynthesis is you have to write 6CO2, carbon dioxide. They can be emit from the stomata. You can see it sometime of the guard cell. There you can see some kind of the guard cell. In the guard cell, you can see stomata. CO2, 6H2O, then you can see here the chlorophyll and the light energy. Then they will form glucose. Then they will form the carbon dioxide, not carbon dioxide. They release oxygen by the photosynthesis. That is why plants are very important in the global warming. When the global warming is happen, there is a temperature increase. What are the main impacts can be from the global warming? You can see it is a climate change, heavy raining, heavy drought, and some kind of the land become a desert, and also the climate change, and the yield is decreasing of a plant yield is going to decrease. These kind of the activities you can see impact of a global warming. So in the environmental chemistry in your last O level part 2 book, that is saying about the what is the main impacts you can see during the high amount of the temperature increase, carbon dioxide form. Mostly that carbon dioxide increasing is the main impact of a global warming. How can this carbon dioxide can be released? The fossil fuel burning, organic burning, incineration, and biogas sometimes release the CH4, methane gas, and some kind of the nitrogen oxide, sulfur dioxide, and nitrous oxide, noxus. We are saying noxus that can be emit various function. Likewise, a wetland. Then that is a very important about the CO2 increase for the global warming. instead of the photosynthesis, yes, carbon dioxide can be observed and emit the oxygen level. That is then decrease, right? So you have to change this equation for the roundup way. It is a cellular respiration. Sometime in your paper, they maybe ask you have to write the equation for the cellular respiration. You have to change the photosynthesis organism to the this one, but you have to use the final output ATP, right? Not ATP. So it is, you can see, it is an overall rod shaped membrane and membrane bounded organelles, aerobic respiration, not anaerobic, aerobic, right? So take place within a mitochondria and release the energy. It is a very important, what is the respiration take place? Aerobic, not anaerobic, right? Aerobic respiration. Aerobic respiration take place, and it is known as the power of the cell. It is known as a power of the cells. Just you can see the Golgi complex. It is the membrane bounded sketch on the top, or the other which is associated secretory vesicles, the collectively known as the Golgi complex. Golgi complex is a membrane bounded sacs, right? You can see some kind of the sacs here. This is a very easy. This is, you can see some kind of the uh, sex, right? So membrane bounded sex stacked on the top of a other with associated secondary vesicles are collectively known as the Golgi complex, but exams not asking define the term Golgi complex, but you have to not, they are asking, but you have to know, right? So Golgi complex function production of secretory substance and the packaging and the secretion. Just you can understand because it can be secretes variable enzymes. It can be emitted some kind of the enzymes, right? It is emitted by various kind of the enzymes. So next one is ribosomes. Next one is ribosomes. They are the small organelles without a membrane and each made up of a large subunit and a small subunit. So we can found freely in the cytoplasm attached to the endoplasmic reticulum. So ribosomes are mostly attached to the endoplasmic reticulum. When you see as an example, this is just imagine that this is a cell and that cell have a, this is a cell, that cell have a nucleus, then you can see some kind of the endoplasmic reticulum attached here. Then if there is some kind of the ribosome present, you can say it is a rough endoplasmic reticulum. It is based on the ribosomes. Mostly that ribosomes help for the protein synthesis, right? What is protein synthesis? You can see during the transcription and the translation, the transcription and the translation, DNA normally protein into protein into just you can see DNA into mRNA, mRNA into protein, DNA into mRNA, messenger RNA. This is we are calling transcription. Messenger RNA, deoxyribo, just you can see DNA mean deoxynucleic acid. So RNA is ribonucleic acid. This is a messenger ribonucleic acid. What is going to do in the protein synthesis? It is we are calling as a translation, right? So this is a function of a protein synthesis. And you can see this is endoplasmic reticulum. There are two types of endoplasmic reticulum, rough endoplasmic reticulum and the smooth endoplasmic reticulum. So the endoplasmic reticulum, rough one attached with the ribosomes. If there is a present in the ribosomes, you are calling as a rough endoplasmic reticulum. The function is transportation of a protein within the cell. Smooth endoplasmic reticulum function is synthesis of a lipid and steroids and the transport them to the within the cell. That is very important. When you come out with the smooth endoplasmic reticulum. Function is synthesis of a lipid, steroids transport them within the cell. Then is a vacuole. Just you can see vacuole. It is a fluid filled with a large organelle found in the plant, surrounded by a membrane. The membrane that is surrounded by a vacuole is known as a tonoplast. That is very important in the vacuole. The membrane that is surrounded the vacuole, that the membrane is surrounded from here, it is known as a tonoplast. The fluid contained, it is known as a cell sep. Just you can see water, sugar, iron, pigment, straw within the vacuoles. So function is maintain the water balance. That is very important. The vacuole is the main part of a maintain the water balance of a cell and support and the provision of a color to the cell membrane by the pigments. So which parts give the color, support, provision to the cell's pigment? It is a vacuole. Sometimes in the exams they can be asked, right? So science is not going to easy paper. It is a moderately hard. Just you can term to understand the basic concept. So mitosis and the meiosis, I have taken here sometime in the paper. They can be asked what is the difference along with the mitosis and the meiosis. Just you can see mito means second, right? Mitosis, just I have added here, this is a mitosis figure. I will do this basic fundamental. Mitosis here, that is likewise a 2n. It is can be a likewise a 2n 2n phenomena in the cell division. But in the cell division meiosis, it is a 2n. You can see it is as a 2n. Then this part's again going to divide as a n part, and this is also n part. This is a called as a meiosis. Just you can see the determination of a differences. Mitosis is a it occurs in a somatic cell. Somatic cells is embryonic cells. It is occur in a germ cells, right? Meiosis occur in a germ cell. Mitosis occur in the somatic cell. And the nucleus divided into only once, nucleus divided into the twice, because I already mentioned here and I draw that nucleus divided by just you can see n, put it a n, and put it a n. This is a structure is likewise here. That is n is going to divided, right? That is phenomena they are saying that. So in the divide, nucleus divide into twice, and the daughter cells, there is a two daughter cells. I already mentioned the n. There is a form two daughter cells here. Two daughter cells are formed, not here. Just you can see four, no, right? So there is a single two daughter cells here. There is a four daughter cells, right? Not two. Just you can see haploid gametes in the there is the n1 n, and you can see how much of the daughter cell form 1 2 3 4 here 1 2, right? So that is they are explaining daughter cells are diploid, daughter cells are haploid, right? So and it is occur more frequently, and we will upload this presentation in the group, so you have to take out the note if you wish. So and it occur a less frequently, and daughter cells are somatic organs, and daughter cells are gametes. So and so this is so here you can see there is only one prophase, metaphase, and an anaphase and a telophase. There is two each phase, which is a subphase and the prophase. The number of the chromosomes are not changed in the daughter cells. Number of chromosomes are reduced in a half. Chromosomes number double of a beginning of each cell division. Chromosomes number is not double. Just you can see know the these kind of the terms, because that is quite advanced for you. Just you can thoroughly note down these things. So I'm going to move the next part is self-pollination and the cross-pollination. So the cross self-pollination is there. You can see this is a flower, this is a stigma, This is a pollen. This is a pollen. This pollen, for example, so you are from Colombo, you are marrying a Colombo girl, or else you are boy, you are marrying the Colombo boy. It is a self-pollination. If you are going to marry from the Trincomalee, Jaffna, Candy from Colombo, you are going to do the cross-pollination. Just example. So that is a kind of the flowers. It is a you can see this is the you can see stigma and this is a pollen. This pollen is going to interconnection. This one is known as self-pollination. The cross-pollination, there is a there is a two flower stigma, this one and this one, and there is a flower that is already have a pollen. That pollen is going to move for the this one or else either this one. This is a cross-pollination. So I think you know the thorough view, and let me give a explanation. Just you can see it is the transfer of a pollen grain and form other to the stigma of a same flower. That is very important. Same flower or another bronze, same plant, right? So it does not require any external urgency. Both anthers and stigmas are mature at the same time. In the self-pollination, that is very important to match the stigma and the pollens, or as uh, anthers are, they want to mature in a proper time periods. It occur even where the flower is close to the homologous offspring. That is very important. Homologous, heterogeneous offspring. This is you can produce in a homologous. That is, I mean, with the same morphological characters, same phenomenons, and that is a look like as same structure, like white color plant that is can be self-pollinated. It can be offspring in the white flower. So when you come of the cross pollination, you can see there is some kind of color change. It is mainly as a cross pollination. So it is the transfer of a pollen. It is the transfer of a pollen again from the anther of a flower to the stigma of another flower born the different plant of the same species. An external agent being the water insect, that is compost, here not required external agents, right? So you are not going to put in the pollen or anthers for the stigma. No, there is a medium substrate we needed, so it is not required. But here need in the cross pollination, exactly need anthers and stigma mature at a different time, and it occur only the flower is open. So if it is open on the cross pollination, flowers should be open and produces heterogeneous. This is a homogeneous. So this is a heterogeneous means, so just you can see XX. This is a homogeneous. This is X and Y. This is a heterogeneous. That means the different color. So mix with the purple and the red, mix with the white and the green, likewise. And method of a vegetative propagation. What are the method you can use for the vegetative propagation? The first one is grafting. That is very important. Grafting, you are during the grafting or a girding. So grafting or budding, we are calling as a budding. You can use as a T budding, H budding. That is very important. So grafting, during the grafting, we are going to compact the both similar tissues of a two stems, right? Like a twin budding. What is twin? Just you can see bulk twin and the body. This one is known as this part. This is the rooting part. Stock. This is a stock. We are calling it as a stock. We are calling it as a stock. This is a twin. We are going to lay out and we have to move it, and we have to tightly tap up into the bottom to top, right? We are tightening up a tape bottom to top, not top to bottom, because we are using the bottom to top, because avoid the water bubbles and the water during the heavy raining season. If we are going to tape our tape by top to bottom, there is going to definitely in the budding junction, you can see some kind of the water, it will be damaged. And you can see some kind of the bulb. It is a natural vegetative propagation system. Natural vegetative propagation. You can see bulb, suckers, runners. You can write suckers. Banana plant have a suckers and runners. Centella, Goticola, we are saying the Sinhala, runners, suckers, runners. And this one is tubers. Tubers mostly consists with the potato and dahlia tubers. You can see some kind of the tubers eye. That eye can be from new plant layering. We can divide as two layering, air layering and land layering likewise. Just you can do the air layering. If you cannot slot it out the stem into the below part or a soil layer part, you can do the air layering. So then in the land layering or a simple layering, you can use the soil mostly. So just I will quickly write a small paragraph. Just you can write this is the how they are doing the uh, this is the main, basically you have to know this is a stem, this is a stem. So this stem you have to remove the from the outer layer part. It is a phloem you're going to remove. So you have to know the mechanism. I'm writing the layering. So this is a leaf. You can see the definitely function. So because this is a remove for the plant phloem, there is no any phloem to see. So that leaves can be do the photosynthesis, right? The leaves can be do the photosynthesis, and they can be produce the carbohydrate. They are carbon source. That carbohydrate move from here. It is come. I will write a red light. So you can see here carbohydrate come, and it is come and it is struck from here because there is no any movement because we already remove the phloem, right? We already remove the phloem. So instead of we are removing the phloem, there is the food coming and it is a storage. After the storing, there is increase of the carbon to nitrogen percentage. Then if there is a carbon to nitrogen percentage increase, there is a suddenly you can see some kind of the roots are came from here. Roots are coming. So you can cover this part using the coconut husk into compost and water and topsoil. You can use topsoil and the water, and you can use 1:1:1 ratio. Then you can add some water. Then you have to make the media, and you have to make it from here. That is a mainly bound part of a layering that is basically very important. And I'm going to finally part the tissue culture. Tissue culture is very important in the science. So tissue culture, a method of a biological research. So when you can see here, the tissue culture is a very method of a biological research, which is fragment of a tissue from an animal or a plant referred to the artificial environment, and which they can continue the serve use as their function. The cultured tissue may consist of a single cell or a population of a cell to wall of a plant of a organs. So that is very important. So during the structure, the tissue culture is we are using some kind of the term explant. It is a derived plant from the mother plant. Explant is the plant which part we are going to use in the tissue culture. So acclimatization, you have to know this term. Acclimatization is we are acclimatize the plant. Acclimatization means we are acclimatize the tissue culture plant for the surrounding environment. During our growth plant, we have to provide some kind of a factors. What are the factors we have to given during the tissue culture room? You have to write temperature, 22 to 27 degrees Celsius, and moisture of an environment that is 50 to 80 percentage, and light intensity. We have to properly give the light intensity that is for the moreover 2000, more or less similar 2000 to 6000 lakhs. So this is the part from the tissue culture. So I think you all are clear about that. So thank you, Hafiz, for your replies, and there Shafia and some of the students are replies I can see. Anyway, thank you very much for the joining today. So at this point too, I'm going to conclude the session. So if you have any verification further, you can wrap up me. So anyway, thanks a lot for joining today. Wish you all the best and have a nice day and good luck for your exams. So, and yeah, thank you. Great work, sir. Thank you very much, Faya, for your kind, valuable thoughts. And it is over to the session. Thank you very much once again. I'm going to wind up the session and good luck for your exams. I am Fazli Ahmad from the University of Sri Chavathanapur. Thank you once again.
Welcome.